saying earlier that I remember when I was a young physician here and then in Boston, and patients used to walk in and say, "Oh, but doctor, you look so young," and then they stopped saying that. And now I'm calling for compliments where you say, "Oh, but you look too young to be a professor emeritus," and I realize that that will only last for a short while, and then people stop saying that too. So it's really, Noel, thank you for inviting me here. It's always a pleasure to come back to the Dome and to visit old friends here at Hopkins. What I'd like to do in the slightly less than 1 hour we have is present predominantly new data exploring the gamut of autoimmune disease, and specifically multiple sclerosis, going from some recent data related to identification of antigen-reactive T cells and B cell repertoire to regulatory T cells, and present some very new data that was just accepted last week related to the genetic basis of MS, where we have identified the genes that cause MS. Let me begin by presenting an overview of what the disease is for the non-neurologist. So the question is, what is the primary driving force leading to central nervous system inflammatory responses? If you look grossly at tissue from patients who die from MS, one sees lesions here in the periventricular region around the ventricles. Here is the pontine region with a dye called Luxil Fast Blue, which stains for myelin, and one can see these extensive lesions predominantly in the white matter, but we now know there are early gray matter lesions in the disease. And if you take these cells and look under, that is, take a plaque and look under a microscope, you see that, again, there is loss of myelin. There's a general inflammatory lesion. There are B cells, there are plasma cells, and there are both CD4 and CD8 cells. In fact, the major model used in the field for multiple sclerosis is experimental autoimmune encephalomyelitis, which is, of course, a CD4-mediated disease. I'll talk about CD4 cells, but as sort of a disclosure, when the neuropathologists look at the plaque tissue of patients with MS, they say they're predominantly CD8 cells and much fewer in the way of CD4 cells in the disease. So what I'd like to talk about over the next few minutes are first, what are the target antigens and what new technologies can we use to identify what the target antigens might be in this so-called autoimmune disease? Two, what are the major defects in immune regulation? And third, what actually causes the disease? And the answer is that MS is a complex genetic disease where many genetic variants lead to disease risk. It's not bad genes, not a bad environment, but perhaps the bad interaction between the genetics and the environment. So first, what are the target antigens? So one can use MHC tetramers loaded with myelin antigens, here myelin oligodendrocyte glycoprotein, to examine patients with MS. And this is Jonathan Schneck in the audience, something we've talked a lot about over the past decade or so. And we've attempted a number of times to do this with different tools. We finally have gotten it to work in collaboration with Bill Kwok and Jerry Nepom at the Ben Roy Institute in Seattle. This work was done by Kadir Radasi, and it's now in press in JI. But basically, we used MHC loaded with a MOG peptide, the 97 to 109. The peptide was modified, so it bound better to MHC, a point that Emil made earlier, that many of these are poor binders. And one can see, if one looks after 1 week of stimulation, that is taking T cells, CD4 cells out of the blood here of a patient with MS with no peptide. There's no MOG binding, and here we have a tetramer binding versus expression of CD25 intermediate cells, and one can see the induction of CD4 cells that bind, that is, of CD4 cells binding a MOG tetramer. These are so-called MOG-reactive T cells. In our laboratory, we're always skeptical of just seeing little dots on the FACS plot, so we perform single-cell cloning of these MOG tetramer staining CD4 cells. Here's one experiment. We have 23 different clones. Here's a stimulation index. These counts were 30, 40 counts per minute. And you can see the majority of these cells were indeed antigen reactive, and they followed classical dose response curves and were MHC restricted. So it gives us confidence that the CD4 cells bonding the tetramer were indeed true bona fide antigen reactive T cells. And here's really the critical experiment. Subjects in MS are in dark symbols. The healthy age match controls in open symbols, looking at either the CD25 negative or CD25 intermediate that is activated cells, comparing here to here, and one can see about a half log to log increase in the frequency of MOG reactive T cells in the circulation of patients with MS compared to controls. And this is really it's a question we've been addressing for many years, and it's the most convincing evidence we've seen to date that there indeed is an increase in frequency of myelin reactive T cells. We also see a modest increase in the frequency of myelin basic protein reactive T cells. And again, per the conversation this morning, we believe there is epitope spreading, but this can potentially be one marker of the disease. Then interested in identifying what the target antigens might be. This is work done by Kevin O'Connor, looking directly at MOG, where he isolated the target antigens right from the plaque tissue. Eluted antibodies from the plaque, ran it on HPLC column, and then SDS page. And when you do that, you see that there are autoantibodies to MOG in the IgG from MS plaque tissue itself. We don't see very much of this at all in spinal fluid sera, suggesting that much of what might be going on in terms of antigen specificity may indeed be going on in the central nervous system itself. More recently, we're interested in looking at the relationship between B cells and antibody. A fundamental question is, what does the spinal fluid represent in relationship to what's in the blood and what's in the brain? We sort of thought that the CSF may be kind of the urine of the brain and may not represent what's going on, but we're interested in examining this in more detail. We created a B-cell receptor library, a transcriptome, using laser capture microdissection of B cells from the plaque of MS patients, did single-cell PCR, generating recombinant antibodies, sequenced the regions, and we spent a lot of time looking at the antigen reactivity of these clone products, and we've not really been able to clearly identify any true autoreactivity. We've also looked at these B-cell aggregates. I just mentioned that an emerging idea in the field of MS is that the earliest event, in fact, are these meningeal B-cell aggregates in the sub-Pia region where Th17, CCR6 positive cells begin, and then they license the classic events of perivenal migration of T cells into the plaque tissue. Much of this work done in the EA model by Federica Salusto in Switzerland. So we wish to ask whether the B cells in these B cell aggregates in the plaque and in the gray matter at the meninges, which appear to be a very early event in MS, whether these B cells are related. This is work done by Novato, recently published in Brain, and basically just summarize these the experiments. If you look at a parenchymal plaque transcriptome versus the meningeal transcriptome and ask, are these related, you can see, looking at these sequences, that the meningeal aggregate share many sequences with the parenchymal infiltrate, suggesting that they arose from the same original clone. That we can look directly at plaque tissue, normal appearing white matter in different brain tissues. Again, one sees the identity of B-cell receptor sequences among these different tissues. So if you summarize these data, there are many expanded B-cell clones that are present in multiple compartments. The dark bar represents shared B-cell receptor sequences from the same germline that have undergone somatic mutation. Clearly, these are IgG and have undergone somatic mutation and antigen-driven response, and we can see the great deal of shared sequences between the meninges, plaque tissue, and normal appearing white matter. More recently, in collaboration with Klaus Domer and Bridget de Obermeyer in his laboratory with Kevin O'Connor, we've asked a very fundamental question in multiple sclerosis. If we take the oligoclonal bands from spinal fluid and run mass spec and have an Ig transcriptome from the same patient, what do the protein sequences of the oligoclonal bands represent in relationship to the brain? Do they represent what's in the brain? And the answer is, in the few subjects we can look at, they do. So basically, the experiment was to do a protein sequencing of IgG in the spinal fluid, taking the oligoclonal bands. We then run mass spec on these oligoclonal bands, and then take these sequences and try to match them with the transcriptome database from the brain tissue. So heroic experiments. And I'll summarize this in the next slide, but basically the blue represents sequences, and here are the transcriptome, that is peptide sequences from mass spec, and you can see the great deal of identity between spinal fluid and brain, sort of summarized here as an example. Here's spinal fluid, lesion 4, lesion 10 from the brain. You can see the clear identity of sequences suggesting that there was a common germline B cell which underwent further somatic mutation in the spinal fluid and brain. So in summary, the MS CNS harbors a network of B cells that populate distinct compartments. There's a relationship between extraparenchymal lymphoid-like tissue and parenchymal infiltrates in the MS central nervous system. The CSF IgG is produced by B cells that reside in the CNS tissue. And this is critical for clinical research because it says that the spinal fluid gives us insight to what's going on in the brain, something we had not really expected. So let me move on to the second part of the talk on regulatory T cells. And I'll start by presenting some very basic immunology related to the functionality of regulatory T cells and show how we've used it to try to understand the immune defects that go on in patients. Just a brief review of the whole field of T cell immunobiology in one slide. Now, you can be a lumper or a splitter. If you're a lumper, there are, 4 or 5 different CD4 populations. If you're a splitter, there are about 10 to the 11th different T cell populations, each one with its own characteristic. But simply, they're clearly Th1 cells driven by IL-12, where Tbet is a major transcription factor. B cells, Th2 cells, which involve in B cell help and immune regulation, driven by IL-4, where GATA3 is a major transcriptional factor. The Tregs, and often we lump together the so-called TR1 cells and the natural CD4, CD25 high cells, but basically the Tregs is identified by FOXP3, by Shimon Sakaguchi, and then the Th17 cells, which use RRC in humans as a transcription factor, which are induced by a number of different cytokine factors. So just to summarize years of work by many laboratories, there appears to be an increase in Th17 cells in patients with MS and an increase in gamma interferon secreting cells. There also appeared to be defects in regulatory T cells, and I'd like to present some new data suggesting a mechanism of action for the loss of regulatory T cell function in MS. I should say that there's very similar data by Jeff Bluestone in patients with type 1 diabetes. So first, some basic immunology on regulatory T cells. Our laboratory were among the laboratories about almost a decade ago to identify regulatory T cells in human based on the work that Sakaguchi had done, realizing that one had to take the CD25 very high cells, CD4 cells, and indeed those cells in vitro models exhibit suppressive function. We had thought that they were terminally differentiated cells, but examination in more detail taking different subpopulations and non-terminally differentiated Tregs is that there is a fair degree of plasticity, and that IL-1 plus IL-6 can induce Th17 Tregs, that IL-12 can induce Th1 regs. So let me show, that's what I'm going to show you now, the data for these observations. First, a number of years ago, in collaboration with Vijay Kutru, we looked at what drove naive and memory CD4 cells to secrete IL-17, and what we found is that TGF-beta plus IL-21 were the minimal requirements for driving naive CD4 cells to make IL-17, and memory CD4 cells, as in mice, required IL-1 plus IL-6. So we then asked whether these conditions could drive the secretion of IL-17 by so-called natural Tregs isolated ex vivo from normal subjects, work done by Gael Bureau, and we found that IL-1 plus IL-6 indeed induces Tregs to secrete IL-17. Interestingly, this IL-17 secretion was inhibited by TGF-beta. One can look directly at the peripheral blood and see that these Tregs tended to be condition with IL-1 plus IL-6 were both FOXP3 positive and FOXP3 negative. Again, we see how TGF-beta inhibited to some degree the secretion of IL-17. And these conditions did not induce the secretion of gamma interferon in these Treg cells at all. More recently, Marguerite Dominguez-Villiers came to the laboratory and was interested in looking at Tregs in terms of gamma interferon secretion. Could IL-12 induce gamma interferon secretion? And this is a sorting strategy that Marga used, where she had very highly purified population of regulatory T cells. In humans, we define them as CD25 high, CD127 negative, and a FOXP3 positive. Note that the CD127 intermediate population in the CD25 high population can have regulatory T cell function ex vivo. If one takes the whole CD25 population here, then CD127 low defines Tregs, but these are also of interest. We believe these are the IL-17 secreting cells. So basically, Marga took these Tregs ex vivo, exposed them to IL-12, and showed that they secreted gamma interferon. So here are Tregs looking at IL-4, that is gamma interferon, IL-4, IL-17, and IL-10. And one can see that with the addition of IL-12, you went from here to here with a very nice induction of gamma interferon secreting cells. We then looked at FOXP3 expression and asked whether FOXP3 is downregulated by the induction of IL-12. So here we have FOXP3 with IL-2 alone, with the addition of IL-12, and one can see that there's no difference in FOXP3 expression with IL-12. We then looked at a number of different subjects. Each symbol represents a different healthy subject in terms of the expression of FOXP3, and one can see there is no difference in FOXP3 expression with IL-12 addition as compared to memory T cells, which of course did not express FOXP3. We then wanted to confirm this is single-cell cloning. We can't do lineage experiments in human by and large, so the tool that we've tended to adopt over the years is single-cell cloning. We take peripheral blood, spinal fluid from any tissue, put one cell per well with PHA, IL-2, or other conditions. And this allows us to look at what an individual cell does ex vivo without the issue of overgrowth. That is, if you have 1,000 cells and you do something, you don't know that one cell didn't overgrow everything in the culture. This allows us to look at functionality at that level. You can see at a single cell level, with the addition of IL-12, you go from here to here, a very nice expression of gamma interferon. We also, in clones, could induce some degree of IL-10 with the addition of IL-12. This is FOXP3, and this is gamma interferon versus IL-10 over here. And looking at individual clones, again, summarizing this data, you can see how the addition of IL-12 induced gamma interferon in the majority of T cell clones. We looked at transcription factors and found that indeed GATA3 was decreased with reduction of IL-12 over here, whereas Tbet, as expected, was increased with addition of IL-12, classic Tbet mediated induction of gamma interferon secretion. And one could look directly at Tbet expression in the Tregs, and this is comparable to naive T cells with induction. We looked at cytokine gene expression Tregs as compared to CD4 memory cells isolated ex vivo by TACMAN PCR. So the closed symbols represent the addition of IL-12, the Tregs versus T memory cells. So again, we see that there is no IL-17 secretion under these conditions with IL-12. Has a nice induction of gamma interferon in the Treg population, no IL-4 induction, again the induction of IL-10. Now, in terms of their suppressive function, we wish to ask, what role did the gamma interferon have in the loss of suppressor T cell function? We wanted to know if there's perhaps a mechanism related to what we saw in multiple sclerosis. So this is a very nice CFSE assay. We basically stopped using thymidine in our laboratory. And basically, if you load CD4 responder cells with CFSE, you have this very nice expression, a loading of CFSE. If you And if you cross-link the T cell receptor with APCs, you enter in the cell cycle with this nice solution. And so we took these cells that had entered in the cell cycle and added back Tregs with the addition of antibodies to IL-10 gamma or combination. And you can see that adding Tregs back totally abrogated the induction entry in the cell cycle. So here the Tregs are able to suppress proliferation in these CD4 cells. If you then add IL-12, you had abrogation of the suppression, went from here to here. The Tregs now were no longer able to suppress. We then asked if you added anti-gamma interferon or combination gamma-10, could you restore the suppressive function? So going from here to here, and the answer is yes. If you block the gamma interferon, or better, both together, you basically almost totally restore the ability of the Tregs to suppress. So this indicated that the addition of IL-12 to the culture, leading to gamma interferon secretion, blocked the suppressor T cell function, which could be restored with blocking antibody. We also looked at the effect of IL-12 on other family members of the IL-12 inducer family, and found that it was predominantly the IL-12 that was able to induce gamma. IL-23 and 27 could not do that. So we then, being clinical immunologists and neurologists, wanted to examine patients with MS, and we asking what the role of these regulatory T cells might be. First, we had previously shown work done by Claire Bacher Allen some 8 years ago that there are defects in regulatory T cell function, the CD4 positive, CD25 positive cells in patients with MS. Here's a ratio of Tregs to T responder cells, patients with MS. Here, age-matched healthy controls. And there's this marked defect in the functionality of Tregs in patients with MS. And this has been replicated by many different laboratories over the years. And the question is, what is the mechanism? So we wanted, again, to ask, what is the mechanism for the loss of regulatory T cell function? So basically, we, in these experiments, Marga took Tregs, isolated Tregs by flow cytometry using the metrics I showed you earlier, right from the blood of untreated patients with MS, stimulated them for 4 hours with PMA onomycin to get them to secrete whatever they're programmed to do. And here's an example of a patient, gamma interferon versus FOXP3. One can see this very nice induction of gamma interferon. Here's another patient looking at gamma versus 17, and one can see the induction of gamma interferon in the MS patient, but not in the control. Here's a series of subjects we looked at, and one can see there was a marked increase in the frequency of gamma interferon secreting Tregs in patients with MS that were not seen in controls. We didn't see any IL-17. That was surprising because we thought we'd see more IL-17 production. Actually, Claire and I spent years looking for it, never could find it. We couldn't because it wasn't there. It was important to ask whether these were just activated cells that were expressing FOXP3. Best way of doing this is looking at methylation of the FOXP3 locus. So here we have FOXP3 positive interferon gamma positive cells, FOXP3 positive interferon negative, and then T memory cells, which make gamma interferon. One can see very clearly, looking at the different CpG positions, that the FOXP3 locus is demethylated in these populations compared to memory T cells, indicating these are bona fide natural Tregs. I should mention this work came out about 2 weeks ago in Nature Medicine. Marga's the first author. We then looked at whether Tregs from patients with MS are more sensitive to IL-12 ex vivo. So if you culture them for 4 days with IL-12, here's an example. Control MS, you can see that there's more induction of IL-12 in the patients with MS, that is of gamma interferon, compared to control subjects. Here's a summary of these data, that IL-12 induces more gamma interferon secretion. We phenotypically characterize these Tregs ex vivo in patients with MS, here controls versus MS, and there's only one surprise really. So as expected, they have more Tbet, more CXCR3, less TGF-beta. But what was surprising to us is there's less CTLA-4 in the cells, and perhaps that also relates to one reason why the Tregs are not working with the IL-12 induction. And finally, wish to ask if we block the gamma interferon in these Tregs from patients with MS, did we restore the ability to suppress? So how important was gamma interferon in mediating this loss of suppression? And one can see that the addition of anti-gamma interferon partially, not totally, but partially restored the suppressor ability, suggesting that this is one part, though probably not the only part, of the loss of suppressive function in patients with MS. Last, we wanted to do the in vivo experiment. Now, if you do mouse work, what you do is you treat the animals with anti-IL-12 and see if you abrogated this gamma interferon secretion. It's not so easy to do those experiments in human, so we took advantage of the fact that beta interferon, a known treatment for multiple sclerosis, decreases the amount of IL-12 secreted by monocytes in the disease. We asked, what does it do? And I'll show you that it decreases the Th1 regs, but induces Th17 regs. So here's basically an example. Here's a control patient. Here's an MS patient on beta interferon. This is a cross-sectional study, and you can see that we've lost the gamma-secreting cells, and now we have IL-17 secreting cells. It's a summary of these data. We have this increase in IL-17 secreting cells with beta interferon. I have no idea what the functionality of IL-17 secreting Tregs are in MS. It's a recent observation. We're certainly trying to pursue this to see what they mean, but reminiscent of recent work by Larry Steinman, so published in Nature Medicine, showing that with beta interferon treatment, you have increase in serum IL-17. So in summary of this part of the talk, we have defects in regulatory T cells in patients with MS, in particular decreases in these Tregs. And I didn't show these data today, been published a number of years ago, but I should mention that there are also defects in so-called TR1 Tregs, that is IL-10 secreting Tregs. Again, there isn't one antigen, there's not one immune defect, but there are probably multiple antigens, multiple regulatory T cells that are dysfunctional, leading to the disease. But let me end with presenting these new data about what actually causes the disease. And along the line of there's not one defect, immune regulation, or not one defect, one type of antigen, MS is a complex, so-called complex genetic disease where there are multiple genetic variants, each with it by themselves a small effect, which lead to disease risk. So what's the evidence that MS is a genetic disease? The population risk is about 1 in 1,000. SIB risk about 2 to 4%, about the same as dizygotic twins. Monozygotic twins have about a 30% risk. Now, Emil made the point this morning, which I'll comment on now, that there's about the same data in type 1 diabetes, suggesting 30% genetics and perhaps another strong environmental component. And I absolutely agree that there's clearly an environmental component. But I think one has to be cautious of saying that it means it's 70% environmental. And that is because we've identified 57 bona fide genes, I'll show them to you in a moment, which clearly cause risk of developing MS. There is almost certainly to be a gene dosing effect. So if you have, and let's say there are 100 genetic variants at the end of the day, if you have all 100, your genetic risk could be 100%. And maybe if you have, let's say 20% of them, maybe your risk is only a few percent, that is among identical twins. So we don't really know with proper genetic loading, with loading of all the risk factors, genetic risk factors, whether in some instances the genetic risk may not be 100%. So 30% represents an average of all the genes, and we can do this experiment looking at concordance and discordance in identical twins and asking, do they carry more of a genetic load? And preliminary data suggests that indeed they do. We've known about HLA-DR 1501. This was discovered back in the 1970s. The naysayers will say, well, you've known this forever. What good has it done? Has it led to any treatments? And I would point out that the answer is yes, that one of the major drugs used to treat multiple sclerosis is sclerotiramab acetate, or Copaxone, and that, in fact, is a ligand for DR2. Now, it's sort of obvious, but in fact, if we didn't know that DR2 and we presented it that way, it'd be a nice story that we made a ligand for this genetic variant found, and it leads to a treatment for the disease. Now, in complex disease genetics, it's very different than the disease genetics associated with so-called knockout diseases. If you have muscular dystrophy, you have a knockout of the dystrophin gene. If you have that knockout, there's total penetrance, and you will get muscular dystrophy, those so-called Mendelian diseases. The common diseases are caused by multiple genes, each genotype having a very small effect. However, the phenotype of the genotype, that is, the closer you are to the actual function of a genotype, can be quite powerful. And I'll show you some of those data in a moment. Clearly interact with the environment leading to the disease. So there are many allelic variants with small but highly statistically significant effects, because we examine many patients coding for disease susceptibility. Though again, the variants have a very major phenotypic effect. So without going through the history of how we got here, we organized some decade ago the International MS Genetic Consortia, led by myself, Steve Hauser, and Alistair Compson, and we performed among the first whole genome association scans by mankind. The results of the first work published as the IMSGC was in August of 2007, where we finally, for the first time, identified risk alleles outside of the MHC region. And basically, because of multiple hypothesis testing, the cutoff is about 10 to the minus 7, 5 times 10 to the minus 7. And we had 2 bona fide hits outside of MHC, the IL-2 receptor and IL-7 receptor. These have been replicated by many different groups. The combined p-value is about 10 to the minus 35 now. So these were the first hits out of MHC, clearly associated with disease. So I'd point out that the actual odds ratio are relatively small, 1.2, 1.17. We performed a second genome-wide analysis. Phil Jaeger in our group at the Broad and at HMS identified a series of other genes outside of the original group. CD58 was replicated, CLEX16A, IL-7 receptor, TNF receptor, RF8, and CD6. Now, we kept going. This was the first GWAS scan I showed you from the New England Journal. This is the second one with 3,000 subjects. And we put together recently a genome-wide association scan with 19,000 subjects. That was done in collaboration with the IMSGC and the Wellcome Trust. And this particular study was headed by Steve Soster and Alistair Compson as part of our consortium. Now, why do we keep doing GWAS scans? And the answer is because you need power to detect a complex phenotype such as disease risk. So the statistical power of 80% at a p-value 10 to the minus 6, you can see for an odds ratio of, let's say, 1.3, for a mean allele frequency here of 0.1, this is 10%, 20%, you can see as you increase the number of subjects, you have a much increase in the likelihood of finding an important variant. So at about 10,000 subjects, for mean allele frequency of 5% or better over here, we're sort of at 10,000 subjects in the sweet spot of finding virtually all or most of the genetic variants associated with disease risk. That is a common variance. This is a consortium, and I'm really proud of this consortium. We basically have gotten everyone on Earth working together in order to do this. We have over 40,000 patient DNAs at our bank at the University of Miami. And here are all the different institutions involved. We started with 19,000 cases, and after QC ended up with genotyping 10,000 with 19,000 controls and ending up examining 453,000 SNPs. The analysis of the data was really quite complex because of looking at the whole world of European ancestry. Turns out the Finns are quite different from the Southern Europeans, which are quite different from Ashkenazi Jews, and so you had to compare people, each group, to each other. And basically, on the next slide, we'll summarize these in a way of looking at it. But this is, and these data are now in press in Nature. This is what I call the periodic table of multiple sclerosis. These represent 57 bona fide, among these 57 bona fide variants, and others are likely to be replicated genetic variants, which are clearly involved in risk to developing MS. They are predominantly immune genes, and really indicate that MS fit into the cluster of autoimmune diseases. So you can use something such as ingenuity pathway analysis. You see immediately how these genes tend to cluster on antigen-presenting cells, Th1, Th17 cells. And at the end, I'll show you some recent data begun to use more sophisticated pathway analyses to take these allelic variants and put them on specific pathways with a very strong indication of an important role of Tregs and Th17 cells. So let me get back to the issue of genotype-phenotype. To me, this is perhaps one of the most critical issues facing us. These data are really for the next generation of scientists out there. We're not going to figure out in our generation. It's going to take years and years to figure out the biologic roles of each of these, but we've begun to do this. And I'll show you some data on CD58, RF8, and CD6. Now, particularly interesting looking at CD6 and CD58. They're both co-stimulatory molecules. And paradoxically, the MS susceptibility alleles for CD58, which is LFA3, and CD6 are associated with lower cell surface expression. Now, that's not obvious. If I asked you to predict what would you expect to see, you would expect to see that LFA3 and CD6 would be increased, that if you have the risk allele, you'd have more expression of those co-stimulatory molecules, right? I mean, we would all predict that. So this begins to give us insight into biology. This is work done with Phil Dieger and others in our group. So here's the CD58 locus. And one little point I'd make is many of the allelic variants we identify will say they hit a gene. But you have to push us a bit more and make sure to keep us honest, because sometimes the allelic variant, the tagging SNP, may be 20, 30 kb from a gene, and that may be the closest gene there, and it has a CD or an IL as number, so we pointed to that. So it's very important to ask as we begin to dissect each of these 100 allelic variants, is the tagging SNP really hitting the genetic variant? In CD58, we have this particular haplotype. There are hotspots of recombination on either side, and we're very clearly able to show that the SNP, in fact, this was the first tagging SNP. We did deep resequencing. This is the SNP with the highest association, and is clearly in one gene. So we know that for CD58. I also mentioned people ask, well, do you know what the SNP does? And let me point out to you another very important point. You have an LD block, a haplotype. There are dozens, if not hundreds, of allelic variants with hotspots of recombination on either side. The genetics are never going to tell us what the SNP, which is the causative SNP. You can guess, but it's up to the biologist to go back and ascertain what the different SNPs are doing on the haplotype. All a geneticist can say is that you have a block, you have 100 SNPs, and they're always an LD, and that haplotype has risk. In fact, I'll bet you nickel that different SNPs will have different functions on the particular haplotype, and there may be a reason why nature chose to have hotspots of recombination to keep certain allelic variants together from a functional basis. Okay, so this is a particular haplotype associated with risk of MS, and we developed a model system with David Altshuler at the Broad. We looked at the level of expression of different molecules on lymphoblastic cell lines that had in fact been totally sequenced by the Wellcome Trust. We simply asked, is there a SNP associated with CD58 expression? And we found that the susceptibility allele, the 230 allele, was clearly associated here with decreased CD58 expression, the EBV cell lines. In fact, this is in Han Chinese. This particular variant is relatively rare in individuals of European ancestry, so it's helpful looking at cell lines from different populations. The p-value was 10 to the minus 10th in terms of linkage. And when we looked at 240 MS subjects ex vivo, we indeed replicated this with arrays that indeed the AA variant was associated with decreased CD58 expression. So then, well, what's the biology of that? Well, we may never know what the precise biology, but it led us to look at Tregs and ask, well, if you stimulate Tregs with the CD3, CD2, the ligand for CD58 versus CD3, CD28, might this be associated with FOXP3? Here, responder T cell stimulated, that is, with an emulator CD58 expression. And one can see that if you look at FOXP3 expression, there's about a log more FOXP3 with stimulation via CD2, again CD58 stimulator, as compared to Treg stimulated with anti-CD28. Indeed, the CD2 locus is one of the nuclear factor binding sites for FOXP3. So it suggests a potential mechanism of action of why decreased CD58 leading to decreased CD2, therefore decreased FOXP3 expression, may be linked. Is this a true mechanism? How do we ever really know? But it leads to some very interesting biology, and more importantly, it can lead to clinical trials. We hope to use anti-CD58 as a treatment in MS based on these data. CD6 is particularly interesting. The particular SNP is on intron 1 of the gene on chromosome 11. It's only one haplotype, so we know it's in the CD6 gene. It's expressed on thymocytes, T cells, and B cells. CD6 co-stimulation sustains long-term proliferation of T cells. And this ligand is ALCAM, which is expressed on B cells, activated T cells, and dendritic cells. Here's the actual locus. You can see the haplotype block. Here's the SNP, the p-value is 10 to the minus ninth, and clearly in this one gene region. So we've created, when I was at the Brigham, this phenogenetic collection taking advantage of healthy individuals. We don't want to look at the function of SNPs in patients with disease. Because if you do, you're looking at all the confounding variables of multiple SNPs and inflammation. So we have 2,000 subjects at the Brigham. We have the collection started at Yale. We have the collection at Cambridge that John Todd has done. And what we've basically genotyped, the thousands of normal individuals have frozen home mononuclear cells, and they come in on a regular basis. We can call in the HOMs who we're interested in and study them. Here's a particular SNP, the C and the G SNP. Here's a population frequency in the Boston cohort. So if you want to study the GG HOM, it's only in 7% of the population. You want to call these individuals in to be able to study them, which we did. Here's a CD6 gene with the multiple exonic regions. There are various alternative splice variants. This particular, the SRC3 region is the ALCAM binding site, and basically the CD6 delta 3 has the ALCAM binding site deleted from it. So we looked at the ratio of full-length CD6 to the CD6 delta 3 isoform and found that if you have the GG variant, that is the MS susceptibility allele, it's associated with decreased functionality, and we splice out the ALCAM binding site. Now, again, the point is, if you want to look at risk to developing MS, yeah, you need 10,000 subjects. If you want to look at genotype, phenotype, you really only need a few dozen subjects. There are likely other genetic variants affecting splicing, but it's pretty clear that if you're the GG, you're splicing out this particular binding site. You don't need that many subjects to examine it. We looked at then CD6-dependent inhibition of CD4 cells. So this is with delta 3, the ALCAM binding site's missing, compared to full-length CD6. And here experiments that work done by David Coughlin in the laboratory. Basically, he's a representative of experiment 1, 2, 3. Here's a CC, CG, and GG HOMs. You can look at entry in the cell cycle blocking CD6. You can see that if you're CC, when you block with anti-CD6, you go from here to here, you block entry in the cell cycle. But if you have the risk allele, anti-CD6 doesn't block. And here's a summary of these data over here. So if you're CC, if you're CG or GG, you have a marked inability to inhibit CD6 co-stimulation or gamma interferon production. We also looked at proliferation, just taking CD4 cells ex vivo from patients with MS and looking at entering the cell cycle in relation to genotype. And one can see that if you're the GG HOM, you have less entry in the cell cycle as compared to the CC or CG. Now, in human immunology, as anyone knows, if you look at any function, you'll have a range like this. And for years, we've been ignoring. If I asked any of you mouse immunologists doing an experiment, would you catch the mice out in the back street in Baltimore and put them in cages and then try to do something with a cage of mice, and they were different species of, you'd say you can't really do that because it's hard to compare them. Well, that's what we've been doing in human immunology. And I'd argue now that we need to know the ancestry whenever we do human experiments, because we need to control for when we see an effect. how much is due to the genetics and how much it is due having mixed mice in the cage, and how much is due to the function we're looking at. So right away we see about 20% of proliferation is due to GG, and we need to do large-scale experiments taking thousands of normal individuals, looking at broad immune function and to find the immune response genes, and to use regression analysis to normalize our results. And basically to look at mechanism, we then said if we knock down this delta 3, the exon 5 siRNA, can we replicate what we find in terms of proliferation? And the answer is yes. Here's a knockdown of exon 5, and you see this decrease in proliferation by knocking it down. So this appears to be the mechanism. So in summary, this A allele of CD58 associated with the increased risk of MS, decreased expression of CD58, whereas the 178 allele associated with increased risk again, and again decreased expression of full-length CD6 with a non-functional delta 3 isoform. And we find that the proliferation gamma secretion becomes CD6 independent in those individuals, again providing perhaps part of the mechanism for disease risk. Just one for the neurologist here, the RF8 locus is particularly interesting. We have a hit at 10 to the minus 9th in the interferon response element region. We looked at 240 subjects with MS, some beta interferon treated, not treated, and asked what pathway does this SNP link to, and does it link to response to treatment? Basically, so this allelic variant associated with the beta interferon response elements, again, beta interferon, the treatment for MS. It's in the interferon signaling pathway. But perhaps most interestingly, if you look at response to Copaxone, genetic variants have no effect on disease. Whereas if you look at response to beta interferon, if you're AA, you appear to respond less to beta interferon compared to the GA or GG. And I think this is really the beginning of what we need to be doing in all human diseases, begin to predict who will respond to treatment based on genetic variation. So the RF locus is associated with widespread effects on type 1 interferon transcriptional responses and may predict response to treatment. The last few slides are, well, how does MS fit into this? And I'm going to tell you about this marvelous experiment being done by mankind to look at the genetic basis of human autoimmune disease. So this is work which is now in Press and Post Genetic, led by Chris Kotsapis and Mark Daly, part of the Federation of Clinical Immunology Societies. And these are the early genetic data looking at genetic variants identified some 2-3 years ago: social autoimmune disease, celiac, diabetes, MS, Crohn's, RA, and psoriasis, and call this sort of the mosaic of human autoimmune disease. The common genetic variations, some going in the same direction, some going in opposite directions. PTPN22 clearly not being found in MS, but being a major factor in type 1 diabetes, Crohn's disease, RA, though it goes in a different direction in Crohn's disease compared to RA. If one does clustering of these multi-phenotypic SNPs by patterns of association across disease, one begins to see very clear pathways involved in disease. So, for example, this IL-23, IL-17 pathway is highly associated with psoriasis, Crohn's disease. Of course, these two diseases are well associated, and multiple sclerosis. The p-value here is 10 to the minus 141. In fact, the disease MS appears most associated with on prospective studies is Crohn's disease. I think provides real clues about disease pathogenesis. There are other pathways which are not involved in MS but are shared between RA and lupus, so-called rupus, and celiac disease, and the IL-2 pathway. So this really, I think, is beginning to give us insight just on very early data. And so the experiment that's being done is the Immunochip. The experiment is now done. It's about to be analyzed. It involves looking at all these diseases with 200,000 SNPs culled from 1,000 Genome Project and all these experiments we've done identifying gene regions with 180,000 subjects. This will tell us what the common genetic variants are among the different diseases. What is shared, not shared. The problem with these types of analysis is it's a post hoc analysis of the different genetic variants on different platforms, different SNPs. But this will allow us to directly compare, and all the genomic calls are being done at the Sanger, so we'll have directly comparable data. isn't it going to be marvelous seeing what that looks like, what genetic variation really looks like? If you see a hit in the IL-23 receptor pathway and you have fine mapping, was there an original sin and something happened leading to that genetic variant? And it'll be the same in all patients? Or is IL-23 receptor particularly important and it kept getting hit at different times? And this experiment will answer that question. So those data will be analyzed soon. The pathway analysis indicates that genes associated with autoimmune disease are enriched for preferential expression Th17 cells. I won't show all these data now, much more than expected, and common interactors are connected preferentially for expression Th17 cells. The last slide, let me just end in a symposium on autoimmune disease is what caused autoimmune disease? Why does it exist? Why is autoimmune disease, MS, type 1 diabetes, Crohn's, so rare individuals of pure African ancestry, not admixed populations like in the US? Why are these diseases most prominently of European ancestry? We know we migrated out of Africa of European ancestry about 50,000 years ago. We know that there was a population bottleneck. If that population bottleneck was Noah's Ark, that's one explanation, but I would prefer exposure to new microbes, but half Neanderthal man in northern Europe, leading to decimation of the population. If the migration occurred in one generation, there are 4 founders of European ancestry. If it occurred over many generations, probably about 1,000 founders. So there's a real bottleneck that occurred. And the hypothesis that many genetic variants with small effects selected for survivors for heightened immune response. So the price to pay was increased risk of autoimmune disease. I just up and acknowledge all the individuals. I think I acknowledged them as I went along, and in particular, Hauser and Compton for the IMSGC. Phil Dieger has been continue to be a partner with lab at the Broad Institute, and David Koffler, Margarita, and others involved in the functional studies I showed you today. Thank you.
With the TH1 regs, as you're calling them. It's kind of a 2-part question, but you probably can answer in one sentence. Is with interferon going up and CTLA-4 going down, could this possibly just be showing cell plasticity and that these cells may be pathogenic in the EAE model, so they're not even considered regulatory cells anymore at this point?
Because I can't speak to them in the EA model, but Vijay Kutra has similar data. Yeah, I think they don't function in MS by losing CTLA-4 and inducing gamma interferon. Of course, there's a lot of data from Sakaguchi, Sharp, and others on the role of CTLA-4 for regulatory T cell function. And we harped on the gamma interferon and what that may do, but I think loss of CTLA-4 may also be part of their loss of functionality. But disclosure, I don't do mice, I just do humans. So. Mice are fun, but they're very important, though. Yes.
Okay, I really like that the talk showed that this, autoimmune disease has many possible genetic effects. But when you start comparing it with Crohn's, for instance, you also got, the other side of the story with the interaction with the environment and all the different antigens present in there. And I know that actually some people think that MS is caused by this viral response, and that the mock-reactive T cell is the basis for that. It's like a molecular mimicry to a viral disease. And I'd really like to hear your opinion about that. What do you think of this?
Yeah, so the question is, people, probably people over here, believe that MS may be due to molecular mimicry with viruses. And what do I think in terms of, let's say, Crohn's disease, and which is clearly related to bacteria. Okay, I was one of those believers up until a few weeks ago. I changed my mind. I think it gets back to no guts, no glory. We've identified a species of Clostridium in the gut associated with Th17 production. We are deeply looking at the microbiome in multiple sclerosis, and if I had to bet my nickel now, I bet it has nothing to do with viruses and it has to do with the microbiome. If I had to bet, but I only bet a nickel. Agree.
So why women? I mean, you've got this beautiful data set
see anything with regard to some of the genetic variation to explain
Yeah.
why women?
And that Y chromosome is so measly. There's nothing
It
there.
is. It's
You
just
guys
here,
know that.
just waiting.
There's nothing there. You know, that's an obvious, a wonderful question. No, we've not had hits on the X chromosome. No. No. None of the hits here. It doesn't. We haven't identified a genetic basis for MS related to the genetics. Interesting. We're probably the strongest genetic factor out there. We certainly have looked very hard, but that hasn't provided an answer. You know, and of course, MS patients, men who get MS, don't do worse than women who get the disease. So, again, it's that measly Y chromosome, I think.
David, thanks very much. Two quick questions. First, the association in the IL-17, IL-23 pathway was between patients with MS and Crohn's?
And psoriasis.
And psoriasis. And so, clinically, how often do you see that? How often do you see that as an association?
So the question, how often do we see the association between MS and Crohn's and psoriasis? The best study to date that I'm aware of, and maybe other, but there's a study done in Denmark, a prospective study, suggesting that the strongest association with MS, and it's really 3% or so, associated with Crohn's disease, in terms of in the same family, concurrence in the same family or same individual. So there is epidemiologic data to suggest that MS goes along with Crohn's disease.
But nothing say as high as celiac and
nothing. Yeah, I mean, clearly the association of lupus and rheumatoid arthritis, and we have a disease which you call lupus, myasthenia gravis, and even neuromyelitis optica, which probably groups there, is much stronger than MS with Crohn's disease. And it's going to be fascinating. We were chatting last night about the GWAS scans being done in myasthenia. And it's really critical for the community to take all these different groups. And you won't need as many patients to do these types of analyses because you already have a prior. That is, you can look at the 300-400 genetic variants, identify those haplotypes. So you don't need as many subjects to look at myasthenia, relatively rare disease in terms of commonality.
And have you been able to look at any of the epigenetic marks in terms of your WASP associations in your studies at this point?
We have had an opportunity to do that? Yes. We've had lots of opportunity.
Lots of opportunity and
no, so what we're doing without being, so we've set up a major collaboration with Brad Bernstein and Rick Young, and we're really trying to look at, are the allelic variants in places of nuclear binding factors for GATA3, TBET, RRC, and other transcription factors. We're looking at very hard. We've taken different, we often go back to basic immunology. So we've done RNA-seq and whole methylation studies on different populations to see if we can cross that with the deep immunochip data of genetic variation, see if the hits, tag SNPs tend to be occurring in regions of methylation. So we're in the process of doing that very deeply.
We'll invite you back.
As long as I get Bertha's muscles.
I want to comment on the other barrier, which is getting from the SNP to the function gene function, and comment on increasing information on the selective value of infectious diseases and how that can be used to determine what the functional genes are. So now we have studies in Africa where malaria is still a major selective agent, and by looking for genes within the SNP range that confer some degree of protection, we can begin to actually identify the gene function of the human as well as mouse.
As my grandmother would say, from your mouth to God's or NIH ears. So we wrote a large U9 team which was funded, Yale, Emory, Mayo, group at Harvard, and looking at response of vaccine grant. And one of the things that we're trying to get them to do is just that, to take all these data and to GWAS everyone, at least to do an immunochip on everyone, because you're exactly right. Right now we're doing in vitro, and we need to do in vivo, pick an antigen immunization, look at the immune response. They give us much more information than doing these silly in vitro things we do. So I absolutely agree.